Hello, what's up, friends? Okay, so let's review protein synthesis. Remember, protein synthesis is made up of two steps. What are they? Transcription and translation. In that order. So transcription first, then translation. So the process of turning DNA code into mRNA is transcription. The process of turning the mRNA code into a protein is translation. But let's dive deeper into both. So protein synthesis is completed by two processes: transcription and translation. The nitrogen bases in DNA code for proteins. Remember, it's not the whole DNA strand, it's just a portion of the DNA strand. So if this is like our chromosome, maybe this section right here is coding for a specific protein, and this section right here is coding for a different type of protein. So we'll only just code that one section. So let's say it's the pink section right here. So we would just code that section of the DNA and turn it into mRNA, not the whole entire DNA strand, just the section that we need. Okay, so blank is the first process of protein synthesis. So that's transcription. And that's turning the DNA code into mRNA. This process is required because DNA cannot and does not leave the nucleus, but mRNA can. And this is important. So when we are looking at our cell, we have our nucleus. Remember that the DNA is located inside of the nucleus, and for humans, we have 46 total DNA strands that are inside of the nucleus. And again, this would be like a longer strand, blah, blah. And we need to get the DNA code or the protein code out to the ribosomes. And remember, the ribosomes can be located either on the rough ER, we'll say this is the rough ER, or free-floating in the cytoplasm. So each of these little dots are a ribosome. So if I were to take that dot and to zoom in on it, it would kind of look like a burger, we'll say. It's two parts to it. And we're going to take that that code. So let's say it's like this section of the DNA. We're going to take that section and we're going to turn that into our mRNA. This is DNA, and we're going to turn it into mRNA. And that's going to be, I don't know, we'll say it's that long. And again, it's going to be complementary to the DNA code. So if it was like A, T, C, G, E, no E's, A, T, C, G, G, T, A, then the code on the mRNA would be complementary to it. So again, if the DNA had A, T, C, G, A, T, then the mRNA would have U, A, G, C, U, A, right? And so then this is the code that's going to leave the nucleus and travel to the ribosome. And then that code will be zipped through the ribosome, right? So that's that first section right there, the first section right there. And it's going to read that code. Okay, but the first part is just turning DNA into mRNA, and that's transcription. So this process takes place in the nucleus because the DNA is inside the nucleus, and the DNA is not going to leave the nucleus. We don't want the DNA to leave the nucleus because the DNA has the code for the protein, and it's safe inside the nucleus. If it got out of the nucleus, there's higher chances that it could get damaged, which leads to mutations, which could lead to different issues like cancers and stuff like that. So we want to keep the DNA safe inside the nucleus. Okay, blank is the second process of protein synthesis, so that's translation. And I already kind of went through some of that. turning the mRNA code into a protein. This process takes place at the ribosomes. Remember the ribs, baby back ribs. You go to Chili's to eat ribs, and ribs are proteins. The tRNA brings one amino acid. So if we go back to this picture, we might have, let's get a different color. Purple. No, it's too close. It'll be yellow. Okay, so we'll have this, we'll kind of maybe make it look like this. This is our tRNA, and the tRNA will have the opposite code, so AUC, and that's going to kind of travel over here, and it's going to link up AUC with the mRNA. So this was our tRNA, and that's going to bring one amino acid. So this would be that amino acid that it brings. And as the ribosome reads the mRNA code, tRNA will bring that amino acid. So it's a specific amino acid that the tRNA will bring. Once the amino acid is brought to the ribosome, then the tRNA will leave, and a new tRNA will come in. So if we get rid of all this, the ribosome will zip in the next three. So we have CUU. So we'll have a new tRNA come in. So that tRNA left. Let's just get a different color. A new tRNA is going to come in that has the GAA sequence. And that's going to bring this ribosome. Sorry. Right? And if we had more letters, then we would have more, I'm sorry, amino acid. This is amino acid brought by the tRNA. So our amino acids will end up linking together at the ribosome. So I'm going to draw it down here, even though it's technically at the ribosome. So we have that amino acid. We have the green amino acid. And let's pretend like our mRNA code is longer. We'll have another amino acid come in, and it could be hundreds of amino acids long. And the amino acid chain is also known as a polypeptide chain, which is also known as a protein. And depending on what the code is, we can make thousands of different types of proteins. Okay, back to the review guide. So the amino acid that tRNA brings depends on the codon. Again, a codon is going to be those three nitrogen bases off the mRNA strand. So the first three, so that would be our codon. And let's just pretend like that's our mRNA strand. And then sorry, my husband just came downstairs and he distracted me, and I'm going to erase this because that's not the right answer. Hold up. Working at home while your husband's also working at home. It's a struggle, you guys. It's okay. All right, let me redo this. Okay, so the amino acid the tRNA brings depends on, I think it's just the code of the mRNA strand. And then a codon is three nitrogen bases that code for, it should say a amino acid. Oh, I guess it should say an. My bad. That's right, right English, right? An amino acid. Okay, during this process, a long chain of polypeptides or amino acids will form, which will make up a protein. Cool. Put the steps of transcription in order. Okay, so the steps. So step one, we know that the the DNA strand needs to separate with the help of RNA polymerase. So we want to go through and try to find which one says that. So step 1, the small portion of DNA separates with the help of RNA polymerase. That's this one. So that's going to be step 1. Step 2, once we have that pocket, so this is our DNA strand, and we'll say it makes that big old pocket right there. And it kind of is opened up like that. And that's going to be with the help of RNA polymerase. So we'll say this is RNA polymerase. Okay, so once we have that, then we can have free-floating RNA nucleotides attached to the DNA template. So let's say the bottom strand is the DNA template, and this is A, T, C, G, A, T. Then we would have our free-floating RNA nucleotides attached to that. So the bottom one's number 2. And let's just make it a different color. We'll say blue. So the ones that will attach to it, if it's an A on DNA, it's going to be a U on RNA, and then an A, and then a G, and then a C, U, A. So that's going to be the formation of our mRNA strand, and that's what's going to end up leaving the nucleus. Okay, so then the mRNA formation is complete at the terminator region. So this keeps coding. Again, remember, if this is our whole DNA strand, we're only coding for a small section of it. So we will start at the promoter region. So let's say this is our promoter region, and we will keep coding until we reach the terminator region. So once we reach the terminator region, then that's it, that the mRNA strand is completely coded for. That's 3. mRNA will then detach from the DNA strand, just meaning it just breaks off from the DNA strand. And so this will come out and break apart. So we'll say it's U, A, G, C, U, A, and we'll just pretend like that's the mRNA, even though it technically would be way bigger than that. And then this, so that's no longer there. And the RNA polymerase will go away as well, and the DNA strand can just close back up. So this will just kind of twirl back up. Okay, so that's 4, and then RNA or mRNA can leave the nucleus. So this will, if this is inside the nucleus, it can leave the nucleus, the mRNA strand. The DNA stays inside the nucleus. Okay, the next one is put the steps of translation in order. So we start off with our mRNA strand binds the ribosome. So once it's in the cytoplasm, it will travel to a ribosome. So let's get a different color. Let's say this blue one is our ribosome. I like to make my ribosome look like a burger. I don't know. Okay, so the ribosome is right there. Remember, the ribosome is the protein factory. So this is where we're making the protein. I'm going to just get rid of this so we have more room to draw for translation. Okay. So once in the cytoplasm, the mRNA travels to the ribosome. Then at the ribosome, the codon of the mRNA pairs up with the anticodon of the tRNA. So let's do some highlighting. So the codon of mRNA is going to be, remember, codon is 3 nitrogen bases in a row. And that's going to pair up with the anticodon of the tRNA. So let's get a tRNA in here. Let's just do this color. And we'll say this is our tRNA. And it's bringing this amino acid. So this is our tRNA, and that's the amino acid it's bringing. And this amino acid is specific to the codon. And so the anticodon, so every tRNA is going to have an anticodon. You could have hundreds of different tRNAs, right? So this specific tRNA will have an AUC, that should say C, anticodon. So let me try to see if it'll highlight in a different color. I don't know if it will. There's like a little box around it, and that's the anticodon of tRNA. Does that make sense? The AUC is the anticodon of tRNA in our example. Okay. Alrighty, so after we have the codon and the anticodon pairing up, the tRNA translates each anticodon into one amino acid, so 3. So that's what this is doing. So we have that amino acid. And then once the amino acids are combined, the codon and anticodon are released from the amino acid chain. So that's 4. So again, we could have already read a bunch of codons. So once this one leaves, so it's like the ribosome will keep the amino acid, the tRNA will leave, so it goes goodbye, and the ribosome is going to zip the next 3 letters in. So if I could try to get rid of all that, and we'll move the ribosome down. So it's like we did that, check. So now the ribosome moves down and starts to read these 3, right? So that's our new codon. We have a new codon right here on the mRNA strand, and we'll have a new tRNA. So this will be a different tRNA because it has a different anticodon. So maybe it's, let's undo that color. This color, we'll say. So this is our new tRNA, and it's bringing a new amino acid, and that amino acid is based off of GAU, right? So that GAU, or sorry, the CUA codes for this amino acid, and these amino acids will link up. So our RNA actually helps the amino acids link together. Our RNA will help link those. So it's like link. And you'll have, again, if there was more coding, so you could have like U, and then you can have another tRNA come in and have another amino acid link up, and you're going to form this long amino acid chain. And that's the last step. So once the amino acid chain is formed, then you will end up with the protein. Now, there actually technically are additional steps after you form your amino acid chain to make the specific protein, but that's as much as we're going to learn in this intro class to biology. Okay, let's get into just talking about some similarities and differences between RNA and DNA. So DNA is double-stranded, while RNA is single. DNA's backbone sugar is deoxyribose. That's where the D comes from. Deoxyribose. Where RNA is just ribose. The backbone sugar is made up of ribose. DNA has the nitrogen base thymine, where RNA does not have thymine. Instead, adenine pairs up with uracil. DNA stays inside the nucleus, while RNA can leave. So RNA is made inside of the nucleus, but it can leave the nucleus. So it could be found in both. What are some similarities? Both of them are made up of nucleotides. Which makes them nucleic acids. They both have the nitrogen bases adenine, cytosine, and guanine, and they both contain the genetic code. And if we wanted to add another one for funsies, we could say both can be found in the nucleus. Okay, All right, well, let's go through our amino acid codon chart. When we're reading the chart, we always are reading off of the mRNA code. So whatever the mRNA is, that's what we're going to be looking at on the chart. So for the first one, we have GUC. So you start off with the first letter. So the first one is going to be G. So we're going to be specifically looking at this quadrant, and we start off with the biggest letter and we work our way down to the smallest. So we automatically are ignoring everything else. So all of these we're ignoring. Don't look at those purple ones. We're just focusing in on the green one. So the G is the first letter. The next letter is U. So that means that we're going to be focusing in on this section. So we now are ignoring all of this. And then the last letter is a C. And it doesn't really matter for this case because no matter what, it could be A, U, C, or G. The amino acid it's going to bring is valine. So that's going to be this one, which is valine. And the anticodon would be for the tRNA that's bringing valine in would be CAG, right? It's going to be coding for that. So let's get rid of all of this and try to do the next one. See if you can do the next one on your own. Well, number 2, you have to work your way backwards. So don't put a CG in there. You need to get your mRNA codon because that's what we're using to look at the chart. So UGC is what we're going to be looking at. So we have our U, then we have our C, then we have our G. So this is going to be the amino acid, which is serine. You hear my dog drinking water. It's actually Cooper's 5th birthday today. It's getting so big. Okay, AUG, when we're looking at that one, we have A, U, G. So this will be the amino acid. Remember, that's a starch codon. Methionine. And the anticodon would be U, A, C. And the very last one, we have C, C, U, which is proline. And that would be G, G, A. Okay, so for the last ones, we actually already did the first one. So this is methionine. G, A, glutamic acid. UGA is a stop codon, meaning we're done coding. So that's when the ribosome will break off of the mRNA strand and stop coding. CCA. All right, and that's that. So that's the end of the review guide. If you have any questions, feel free to email me, text me, whatever you want to do. Your test is tomorrow. So again, feel free to ask any questions. I hope you guys have a great day and love ya. Talk to you later.